Another one that is critical is magnesium. And the lower image there is showing the magnesium deficiencies over time. And with the left leaf, the normal looking leaf, and then you can see how it can get dramatically worse if left untreated. Magnesium is definitely needed in large amounts. And if you have too acidic of a soil system, or if it's a sandy soil system, you will get deficiencies, kind of similar to what we saw with potassium. Potassium, especially in sandy soil, can be a problem. It is actually part of the chlorophyll molecule. So if you just think about that alone, how much chlorophyll a plant makes, it makes sense that they need a lot of magnesium. And then factor in, they also use it for all these other things. This is not a complete list of everything that magnesium is needed for in the plant. It's just some of the major. Rubisco, you see the word Rubisco on that list. That is, of course, the enzyme that kicks off the C3 cycle. So the plant is busy using Rubisco, and they think, when they do the math, Rubisco is the most abundant protein in plant leaves. And so it is definitely needed in high amounts. It's actually needed for ATP as well. I'll show you an image of that in a second. But one of the classic signs is intraveinal chlorosis, where the veins stay pretty green and the tissue around the veins tends to bleach out and go chlorotic. It's mobile in the plant. So that means the older leaves will show the symptoms first. So the right image is chlorophyll. Mock-up of chlorophyll A and chlorophyll B. You just replace that R group, get chlorophyll. So this letter R up here is just saying if you have a methyl group there, it's chlorophyll A. If you have an aldehyde group there, it's chlorophyll B. So we say that magnesium is chelated in the center of this. They call this the head region, the porphyrin head region of the chlorophyll molecule. So got to have it in order for chlorophyll to function. And in fact, one of the electrons in magnesium is the electron that actually gets excited and boosted off during photosynthesis. So absolutely critical. And then I didn't know this until into my adult life. I must have fallen asleep during this part in human phys class, but I didn't know that ATP needs magnesium. And so we all run on ATP. And so again, another reason why we have to have magnesium in our diet. So yeah, I did not realize that. So when you look at agricultural soils and the mineral content of agricultural soils, there needs to be a lot of magnesium. And I'm not super confident that that's the case. So this is one mineral that I actually take supplements for on a fairly regular basis. But you got to be really careful because if you just buy like the cheaper stuff off the shelf, it's usually magnesium oxide, and that stuff you can use it if you want, and it works really well if you want like a laxative effect. If you want to make yourself more regular, go ahead. But if you want to have it so that it makes the magnesium bioavailable to your cells, that's not the form you should get. Of course, they don't tell you that. So you look on the bottle and they don't say anything to that effect. So you got to do your research. I buy chelated magnesium glycinate, magnesium citrate. So I buy the chelated version because they claim that's the version that when you take it, it goes into your cells. It actually can make it into your cells and be physiologically active. You may hear the term bioavailable. That's kind of a buzzword in nutrition industry. Is it bioavailable or not? And you want it to be bioavailable for your cells. So just be careful about that. Okay, iron is a big one. So this is technically a micronutrient, although there's been some debate by plant physiologists that it should make it into the macronutrient category, but I don't think that will ever happen because those lists got formulated so long ago and it's pretty set in stone. But of all the micronutrients, iron is definitely the most, they need this the most in terms of sheer quantity. And you see that I've written down two forms, Fe2 plus and Fe3 plus, okay? And in the 3 plus, I put in parentheses. What I'm trying to show there is that the Fe3 plus form of iron is less available to the plant. And so Fe3 plus, another way to talk about that is to call it the ferric form of iron. And then the Fe2 plus is ferrous form. It's just the OUS on the end. FERROUS. So Fe2 plus is more available in the sense that it's more soluble in the water. Okay, so it's more soluble. Ferric is virtually insoluble in water. In fact, I've worked with ferric when I make up certain recipes to grow my bacteria and fungi on, and I have to include ferric. It's a pain. I always have to include some other chemicals to make it dissolve. So iron is very helpful to the plant because it's involved in the synthesis of chlorophyll, so that's hugely important. And it also plays a lot of different roles in these so-called redox reactions. And generally when they say redox reactions, what they're thinking about is electron transport chain ideas where you're transferring electrons between molecules. Okay, so redox reactions. So they're important for that. They help latch onto electrons and pass them down the line, so to speak. Another, you can get this confused with magnesium if you're not careful because it also has intraveinal chlorosis. However, it's immobile, whereas magnesium does intraveinal, but it's mobile. So here's a good example of just look for where in the plant body are you seeing these deficiencies, and that's how you want. So intraveinal, but it's immobile, so that means the newer growth gets hit hard. So that's what you're seeing here with this plant, where the newer growth up top is showing that intraveinal chlorosis. One thing to be aware of in really acidic soils, it can actually create a toxic effect on the plant, where you may have heard your mom or your dad say too much of anything is bad for you. The same with this mineral elements. They found that if you get too much of a certain element into the soil, it can actually cause problems, not only directly because of that toxicity of that particular element, but then it can also make other minerals less available because of their interactions. So you got to really be careful with that. So toxicity in really strongly acidic soils, but that's usually not terribly common. It's more common that you'll have a deficiency. And that is because a lot of the iron in the soil is unavailable. It is ferric form. And so, like I said, that one is just really, really hard. It's not soluble. And then if it's complex with other molecules, it'll be precipitated out. So very often, if you're going to add iron into the soil, you do a chelated version. And plants have actually evolved mechanisms to do this as well. And so when I work in the lab, that's also what I do is I use chelated chemicals, or they're called ligands. I use chemicals to essentially latch onto the iron and make it more soluble. And plants do this too. So you can, in the lab, like I use EPA when I'm trying to dissolve iron, but plants do their own version. And so they have their own chelating agent like cafeic acid. And so this is something the plants make in their root tissue, and then they send that out into the soil. And the cafeic acid will actually latch onto the iron. And when it gets to the surface of the root, the iron gets cleaved off and brought in. And the cafeic acid is really effective at, it's kind of think of it like it's scavenging for iron and then latching onto it. There's also a group of plants that produce, in general, they just called phytospores. And it's I'm not going to go into details, but this is another class of chelating agents, essentially, that certain plants make. And so phyto, anytime you see phyto, that means it's plant or plant-like. Siderophore is, so plants make phytosiderophores. Bacteria, for example, make siderophores. And so bacteria were I think they were the first to be discovered to have this ability, but basically bacteria make these siderophores that latch on to iron. Well, then they discovered plants do this too, so they named it phytosiderophores. And that is also something that they release out from their roots, latch on to the iron, chelate the iron with it, and then bring it back to the root and allow for the iron to get cleaved up. A lot of the bacteria that infect you, like pathogens that make you sick, they're often siderophore producing bacteria because bacteria need iron, especially pathogens, they need iron. And so they're going for the iron in your body, and they'll use siderophores to do that. And that's one reason, like when my boys were little and they pop a fever and I go to the doctor said, just let the fever run. And it's a little nerve wracking as a mom. You're like, oh, really? You want the fever to just keep running? And they said, yeah, because when you pop a fever, it actually stimulates the production of protein in your body that latch onto iron and keep it away from the pathogen. I'm like, oh, that's brilliant. So that made a lot of sense to me. There's a limit. You don't want a high fever to go too long, but a moderate fever, they said, is actually good for you because it kind of sequesters the iron away from the pathogens trying to get to it. And then there's another molecule that certain plants produce called DTPA, again, just chelating agent for iron. So you can see that there's different chemicals, different species. So this is like a thing. Plants have needed to grapple with this issue, and they've come up with different solutions depending on where they live and their species. But just trying to point out that this is something that's fairly common, and plants have needed to evolve mechanisms to deal with it. Here's just a visual if you are curious about this whole idea of chelation. I'm not going to go into the chemistry of it, but you can see the before and after idea here with the DTPA showing the chelated iron. So iron is very, very important. All right, so that is what I'm going to get into as far as the mineral nutrition. So through this slide set, we've been looking at this idea of mineral uptake, and that, for the minerals needing to be in solution, right? So they need to be knocked off the soil particles, so to speak, and get into solution, and then the plant root can take them up. And so the plant roots themselves have very limited capacity to work with anything that's still stuck to the soil particle, any of those minerals. But remember, the bacteria and the fungi are quite adept at that. They're really good at being able to knock off, solubilize, make things go from precipitated to soluble. And so thinking about the bacteria and the fungi in the soil, they're absolutely critical for the plant to get access, especially when you start thinking about some of the soil systems that are like just overworked. There's not, a lot of the minerals have been mined out of there year after year after year of ag, growing plants in these agricultural soils. And so if you don't replenish that somehow, you really need to rely on the microbes there to help get access to scarce minerals. And so as you grow plants in a soil system, boy, you really need to know a little bit about your soil and also about the microbes in the soil, and that you want to cultivate the right kinds of microbes, right? Because there's definitely there's some good, not so good ones that you want to keep your plant away from. Also, please spend some time looking at biological nitrogen fixation. Okay, we spent a few slides, talking about this idea. I will be asking about that. This is a huge thing in the commercial ag growing system. Looking at, we already have plants that we use for food production and oil production that are forming this relationship with bacteria, and then they're trying to make other plants have this ability. So this is something that is not going away anytime soon, this interest. So, and I think rightfully so, what it can do for the plant is pretty profound. And so make sure that you're clear on the idea here with not just the bacteria and fungi that normally hang out, but this very special case of bacteria hanging out in the root tissue in those nodules. Any questions on anything in this slide set? Okay, well, we're going to stick with the soil. Can't help myself. We're going to do that again. We're going to get into soil management. Now, this is based on one of the chapters in the book, but I'm going to pick and choose the topics in this section. And so, you know, and of course, I'm going to be focusing on like agricultural soil systems, both field-based, but, you know, also people who are thinking about, you know, like indoor ag or controlled environment ag, you still need to know a little bit about soil because some people still use greenhouses with soil-based growing systems. It's not all hydroponic. And so thinking about this, but I'm going to kind of zoom in on more on the ag-based, field-based idea here. And I'm going to be, you know, truthfully scratching the surface on this because there's a whole lot to it. I think farming is one of the toughest jobs out there. It's amazing what you have to deal with on a day-to-day basis. That's so much out of your control in terms of Mother Nature and all of that being thrown at you. And then just the sheer complexity of the soil system is amazing. And so trying to get it right, it's a lot of work. So, you know, if you are buying a plot of land and you want to grow crops, how do you approach it? I think if you just think about it for a little while, you would probably come up with a lot of what's on this list here in terms of, well, you probably don't, ideally, you don't want a really hilly area to grow your crops because then you have runoff. They now know that, you know, synthetic, inorganic, mineral nutrients, feeding that to the soil over and over is not always the best strategy. Organic materials need to be put into the soil system on a regular basis. Tilth is a fancy word that they use, but basically they're just saying the physical condition of the soil composition, the soil aggregates, the soil pore sizes, all of that. Obviously, you don't want weeds in your fields. If you're trying to grow corn, all you want is corn. You know, at least in conventional agricultural fields, that's what they do. They don't want other, even if a soybean plant pops up, but they're trying to grow corn, that soybean would technically be a weed. Obviously, you're trying to minimize disease costs, and erosion, as we know, is not good. So tillage, you hear this word a lot. And basically, it's just the physical manipulation of the soil to try to get it ready for growing crops. Lots of different machinery out there to accomplish this. I am not going to go into all your book chapter does go into it. I am not going to expect you to know all that. But the basic idea of tillage and that there are different strategies, different machines out there to go either super aggressive, where the tillage is pretty profound disturbance, to a fairly minimal disturbance. And then I'll also mention no-till. There's a big push right now for no-till agriculture. We'll talk a little bit about that. But basically, whatever strategy they're doing, they're trying to get at this idea of preparing the soil for plant growth. So you want soil that has the ability to hold on to water without waterlogging it. You want a good pore space ratio. And so the general rule of thumb is outlined here. You want the soil to be 50% solid, 50% pores, and then of the pores, you want half of that holding air at any one time and half of it holding water at any one time. So basically, you don't want a waterlogged system where all the pores are full of water. That's not good because then the roots can't breathe. It's very hard for oxygen to move through the water in the soil system, and so then the plant roots start to asphyxiate, essentially. They need oxygen. So all of these are important things to consider, and what machinery you use to get that to happen depends on what plant you're growing and quality of your soil. When you look at just the soil surface, that can be challenging, because if you have big aggregates of soil, clods of soil, that just means big clumps of soil, that's not good for initial seed planting, because it's too uneven in essence, and you'll probably get a lot of fail to germinate situations going on. So you don't want big clods. And then you need the soil loose enough so that you don't have issues with root growth, right? The roots have to be able to make their way through the soil. And so the subsoil also has to be in a composition where, yes, you have good pore space, and you can't have any compaction going on deeper down. And certainly adequate drainage is important as well. And so in the subsoil, there's a condition that happens in actually, there's a lot of states in the Midwest area. I was just reading quite a few readings from Kentucky area where this is a huge problem. So they give it a special term. It's called the fragipan. Kind of a neat word, fragipan. So the fragipan is actually this super compacted like layer of soil, but it's not at the top. It's further down in B horizon, they call it, the B horizon of soil. And so it's further down than the A horizon. And they're not exactly sure what causes it, but basically it's a super compact layer of soil, so compact that roots can't get through it. Well, obviously that's not good. If you have roots only growing so far, and not, that's as far as they can go, that's terrible. And their ability to then access water and minerals is greatly curtailed. So trying to figure out a way to deal with this, especially when it's deeper down in the B horizon, this is tricky. And much easier to deal with stuff at the surface. You can, use special machinery pretty well to get that. But the fragipan idea, so there's a lot of research going into how do we deal with this? How do we remediate it, essentially? And there isn't a solid answer yet on that, although they're looking at a few different options. So you don't want the fragipan situation if you buy a plot of land. So make sure you always investigate your soil system pretty intensively before you opt to grow any plants there. Okay, what else can you do to prepare the soil? Well, fumigation used to be a tried and true practice. It's less so nowadays for good reason, although it's still being used for certain crops, certain fruits, certain vegetables, and even some ornamentals. They will go ahead and fumigate the soil. So they're using chemicals. It's usually, well, not usually, often it's a gas that they'll pump into the soil. And it's to get rid of any pests or pathogens and any seeds that they don't want germinating. Methyl bromide was the tried and true thing, and was used with great abandon for many, many, many years. Then they realized, oh, gee, it's hurting the ozone layer. Oh, gee, it doesn't have the best health implications for animals and other organisms that interact with this chemical. So methyl bromide has been banned for most plants, but unfortunately there's still some exemptions. They've turned actually to grafting. I thought that was interesting that they're getting into grafting as a way to deal with some of the issues with certain pests and pathogens with the phasing out. So usually what they do is the USDA will phase out these fumigants and pesticides and herbicides, and then they'll say, by the year such and such, it's going to be totally banned. And then everyone is scrambling to find another way to deal with whichever pest or pathogen is in their system. And so grafting is turning out to be a good option. And so getting a rootstock that is resistant to the disease seems to be a good trick. I'll talk about some other methods too. Pasteurization is something that I routinely do when I'm running experiments where I'm spiking in my beneficial bacteria. I will pasteurize the soil to reduce competition. Usually in the initial stages of research, I would opt for that strategy. And that's much gentler because all you're doing is heating the soil and it's a mild heat. You can use an autoclave, which is more drastic. I tend not to do that. I tend to do, you can buy, like, I've purchased a couple different sizes of pasteurization units. And the small one that I have is really simple. It's just a square metal box that has a heating element through it. You fill up the soil and plug it in. And so it's kind of analogous to your oven in your home kitchen, except it's a little fancier with those heating elements throughout the whole box. And then what you should do is run it for roughly 30 minutes at about 82 degrees C, and then let it cool. And then some people, that's all they do. And it's okay if you stop there, but I prefer a second run. And the reason I do another run, I let it cool for a day or two, and then I do another run of pasteurization because you don't kill everything the first time around. And so often what you end up doing is stimulating spore production or spore germination of the fungi or bacteria. And so you might get yourself into a quandary with that. So I always do one cycle run, let it cool. If there's any spores that are germinating, after a day or two, they'll be fully germinated and then they're more sensitive. Hit them again with heat, and that knocks down those bacteria and fungi that germinated off. And that tends to do the job. So pasteurization can be done. I think you have to be really careful, though, of asking yourself, should you even do this to begin with? And now, like I said, I do it when I'm doing experimental research, where it's like, the first run through on this experiment, because it knocks down competition and you get to see how your microbes work in a soil system just in general. But doing this for growing crops in a more realistic setting, pasteurization and fumigation to me doesn't make a whole lot of sense, because you're knocking down, like when I pasteurize the soil, I can't say, now heat don't hurt the good guys, only hurt the bad guys, right? I mean, you're killing everything. And we now know that you need microbes in the soil. I just got done telling you that with mineral nutrition. And so I think either of these methods is not really the best strategy. In certain conditions, it can be useful. But as a general rule of thumb, I would think that this would be not a good idea. So if you look at soil systems around the world, there's all different kinds of quality. And degradation happens over time. And I think, again, if you just think about it, that this list would be something that you could come up with pretty readily. Erosion, of course, is a big one. And, with the Dust Bowl that happened in the early 1900s, where we had severe erosion and where the soil actually went off into the ocean. It was a big historical event and terrible. And so erosion can be a severe problem, but for the most part, we've learned our lessons on that and have, current farming practices are pretty good at thinking about this question and minimizing loss of the surface soil. So, but you can also just in general lose organic matter over time with intensive use of that soil system to grow crops. You can just have, as well as the minerals too. Again, when you grow crops, they're taking up the minerals. And so if you don't figure out a way to replace those minerals, you have an issue. And then salinization is another issue. And again, you don't have to be necessarily near an ocean where there's salt water for this to be an issue. It could just be that you've put too much inorganic fertilizers in the soil. And then over time, they accumulate, and often through capillary action, they'll rise to the surface and start messing with the plant's ability to take up water in particular. And so Again, you don't need to be near an ocean for salinization to be an issue. Compaction is a real problem that sometimes, like if you're not a grower of plants, you wouldn't think too much about this, but just the machinery used on the fields is troublesome. Sometimes that can cause compaction, especially if the water, if the soil is too wet, and then you have people driving over that. It can cause compaction, and obviously that will create much smaller pore spaces. Water has a more difficult time getting into the soil and going to any depth. Roots have a harder time growing through it. And so compaction is a huge problem, including the frangipanidae. All of that is a problem. On the right, that picture that you're looking at, you've probably seen images like that in a variety of different places, but that soil compaction like that, soil crusting at the surface, can be a real problem. And it's actually through rain action, which, you want rain, of course, but it can create these aggregates under the right conditions and really cause problems. And then, it's like, okay, how do you get that loosened up? You're going to have to use some kind of mechanical disturbance to get that loosened. Okay, and then last on the list there was waterlogging. So if you can imagine, especially when you have compaction going on, that soils can get waterlogged more easily under those conditions, obviously, where, the water will seep in, but the smaller pore spaces, so then the soil will get waterlogged. Right? And so now I just wanted to talk to you a little bit about that, because there's some microbiology going on. This isn't just there's too much water and therefore not enough oxygen is getting to the roots issue, although that's an issue, right? So if there's waterlogged soil, that means oxygen is going to have a much tougher time diffusing to the roots, because oxygen and doesn't like to diffuse through water. So you certainly have the roots starving of oxygen, but it's complicated because you also have an anaerobic soil. So hang with me here. So anaerobic means no oxygen, right? Well, if you create a soil system like that for too long, what happens is the bacteria that love anaerobic conditions start to thrive. Plants in general don't like to hang out with anaerobic bacteria. There's some exceptions, but in general, not such a good thing. So what you're seeing here in this image is kind of similar to what we do in our mitochondria. This left image here, where you see that basically what they're trying to show here is an electron transport chain process with oxygen as the final electron receptor. So what this is trying to point out is if the bacteria or the fungus is using oxygen, it's kind of analogous to what's going on in our mitochondria, okay? And you get, in this case, when you think about what's going on in our mitochondria, this sets up a situation where we can get ATP produced. Okay, great. On the right here, we're comparing it to, instead of oxygen sitting at the end, it's NO3 minus, that's nitrate. Plants love nitrate, remember? That's one of their favorite ways of taking up nitrogen. So here you have bacteria or fungi, primarily bacteria, in the soil grabbing nitrate for its own uses. That's not good, because plants need nitrate. But if you create an anaerobic environment, now instead of oxygen sitting at the end of this electron transport chain, you have nitrate in this particular case. So now when the bacteria runs this reaction, it's going to stick nitrate at the end of this electron transport chain and convert it in the process to nitrate. You don't want that either. So you don't want that going on in the soil, because now the bacteria has used nitrate for its own benefit, created nitrate, which isn't the best. Okay, so you start to get this situation where the plant is getting reduced nitrate because of the bacteria hogging it all for their, personal needs, for their physiological reactions. So this, and then if you can imagine a bunch of these bacteria in an anaerobic state, they start to increase their populations. So you can really get a significant loss of nitrogen in the form that the plants need. So some bacteria in a waterlogged situation are going to do something like this, where what I just showed you in that previous image was nitrate getting an electron and getting reduced to nitrite. Well, then other bacteria take it even further, and they'll keep running those reactions where they might produce nitric oxide, nitrous oxide, dinitrogen. Those are all gases. So now you have nitrogen gases, those get released out of the soil. So that's terrible for the plant because you want nitrogen, nitrate in particular, in the soil. And so the bacteria, these anaerobic bacteria, are working totally against what the plant needs in terms of their nitrogen. And so again, if you have waterlogged soil for too long, you create high populations of these types of bacteria. And knowing that and being able to deal with it appropriately is important. Now, the other thing too, so you get loss of nitrogen, but that's what we were just showing. But you get some other issues going on. So other anaerobes, they're in anaerobic conditions, then they start acting on the ferric form of iron, which I just told you is pretty insoluble. So they'll run a little reaction and they'll take ferric and in that reaction turn it to ferrous. Well, then you actually get toxicities building up. You get too much iron available to, like all at once. And that starts to create toxicities for the plant. Some bacteria will take sulfate and reduce it to hydrogen sulfide under anaerobic conditions. That's toxic. Hydrogen sulfide is toxic to any living organism, as well as, to us, as well as to plants. It's a respiratory poison. So that's not good. And then bacteria and fungi will produce other weird chemicals that they'll excrete out that are not good for the plants. So butyric acid is just one example. Right? There's others. Acetic acid too. And so again, it's just getting this shift. Once you waterlog a soil, it's not just the idea that the plants are getting starved of oxygen. It's all the microbes that are doing the anaerobes that are doing their thing. And so this can be a real problem. Some plants, by the way, when they are, if they are relatively regularly hit with pretty waterlogged soil, and then it eventually drains away, and then they live their life, and then they get another huge rain event. They've actually adapted where if you look at their root tissue, they actually create huge air gaps in their tissue, like just dead spaces in their tissue, so that when the oxygen comes into their roots, it's so much easier to get diffused to all their cells. And so plants can actually shift their root morphology in terms of how airy their tissues are to deal with it. Well, they're quite amazing in trying to deal with it themselves, but they don't have a ton of control with all these anaerobes that hang out in the soil with their roots. This is a whole other ball of wax, so to speak. Okay. So it's been really exciting for me to see how people are really starting to pay attention to soil health and understand how complex it is. And if we can better understand it, we should be better at growing plants, nice, healthy plants. And so the idea of what's going on in the soil has finally garnered enough attention out in the real world where people are really doing a lot of research on this. I like this image because I think it hits the main points when you think about soil health currently, what they're trying to do. And like that big yellow one up top, minimize disturbance, that's a relatively new idea. In previous farming practices, that was not on anyone's radar. And now it's a big deal. And I'll explain a little bit more about that. Maximize living roots. Very important that we have good root systems. Soil cover for a variety of reasons, keeping some soil cover. And then diversity, biodiversity. Now, if you've taken any kind of ecology class, you know that pretty much any ecosystem you look at, they point to if it's a diverse ecosystem, it's probably pretty healthy. And that a reduction in diversity usually harms that ecosystem. And whether you think about a forested ecosystem or the ecosystem in your gastrointestinal tract, they're saying the same thing. You need diversity. And so it's interesting that they included that on this. This is from the USDA website. So that was good that they're paying attention to the ecology. It's finally sinking in. Okay, so how can we improve some soil conditions? So cover crops are a hot topic, all right? And so cover crops and green manure. I remember the first time I heard green manure, I'm like, what? Cow manure that's all green? I did not get it at first. But what they mean is green manure is you grow plants in that field and then you tend to till it into the soil, yeah, as a manure. They call it a green manure. And so there's cover crops and green manures are kind of the same thing. It just depends on how far you take it. Because if you take that cover crop and till it into the soil, then it's more appropriate to call it green. So they've been looking at different cover crops, and this is taken from a website that from Minnesota actually, and they are looking at different crops for the Midwest area. And so I like the list that they've generated on these different crops that can be used as cover crops. And you know, with winter wheat and winter rye, for example, you actually plant them in the fall and let them go, and then they'll sprout up in the spring, and then you can till them in as a green manure if you want. At sometime in May, usually in this area of the world. Where is the... And then they're showing some, you know, things you have to be aware of. The root system might be a little too robust in some cases for the next crop coming. But basically, these cover crops and green manures, it's a way to replenish the soil of some good organic matter. And that you want good organic matter because it feeds the microbes in the soil. That's really the big picture idea. You want those additions of organic matter so the fungi and the bacteria can break it down and release that, recycle it, and get that those nutrients to the plant. Soil structure, just getting the physical structure of the soil in the right condition for plant growth. You do need a mixture of organic matter for that. And then it does prevent erosion big time. If you have plants growing on the surface, it really does help with stopping. And so you can, like some people say, well, why don't you just add a bunch of organic, like dead organic matter into the soil? And you can do that, and that is done, but you have to do it repeatedly and frequently. And so depending on your cost, it might be cheaper just to grow a cover crop and rather than dragging in a bunch of manure and putting it in. It just depends on, you know, what plants you're trying to grow and the condition of your soil. Both are options. They're good options, but there's some pros and cons. This is from that same website, looking at the University of Minnesota Extension Service and just looking at different crops. So again, I'm just trying to show you that there's different cover crops that are like a short list of popular cover crops, and these make those lists. And so I've highlighted that third bullet up top because one of the things you have to be careful about is if your cover crop inhibits the next crop coming in that you're trying to grow for a profit, then you got to be, obviously that's not a good idea. So you have to know a little bit about the cover crop. You can't just willy-nilly grab anything and call it good. And so chemicals released by ryegrass may keep small seeds from germinating. So you don't want carrot and lettuce. Yeah. Yeah, and that's something that's just kind of, we're scratching the surface on that idea, the research on that. But certainly what plants you plant, what cover crops you plant in that soil will definitely influence the microbiome of that soil. Absolutely. And then you got to question, okay, if this cover crop alters the microbiome, is that good or bad for the next crop coming in to talk about? You know, because cover crops are usually something you, well, I mean, you could make a profit on, but sometimes they're just manured in, you know, the debris for. So, but yeah, that's something, and we're now realizing relatively recent research is showing that plants have a pretty profound ability to influence the microbiome. We used to think that plants just were at the mercy of the bacteria and fungi in the soil, and they have control on it. Now we're realizing they have a lot of control on what they excrete out of their roots. They actually can attract certain types of bacteria and inhibit others, and fungi. So it's way more dynamic, and they have way more influence than we thought. And so it's really exciting to think about when you think about the plant microbe interaction. So again, you know, I'm not asking you to like memorize these different qualities. It's just kind of some of the big picture ideas of you have to be thoughtful about the cover crop. You can't just pick any old thing. And then of course it depends on the geographic region, which of these would work best, obviously. Clovers are really popular because they are legume type plants where they can um, have nitrogen fixing bacteria with them. And so that's a huge benefit if you can have a cover crop or green manure that associates with nitrogen fixers, then you don't have to add as much nitrogen. Where is the word? Where did I put the word? I wanted to make sure that it was in here. Allelopathy was in here somewhere. Did I miss it? Is it? So that's the other thing too, is, you know, I highlighted the yellow because that's getting at where some plants release chemicals that actually inhibit maybe the next plant you're trying to grow in there. So you got to be careful about it. Okay, I'll continue on with this thought on